Thank you, Mr. Chairman. Can I have the first slide, please? Okay, we'll start my presentation with a short story. We were here in the year 2000, let's see. Yeah, in 2009, there was a meeting here at the casino on astrobiology. In that occasion, I presented my work with extremophiles that we isolated from the Atacama Desert. And the last lecture in that meeting, according to the program, was going to be given by Paul Davis, that you can see him there. I'm sure all of you know him. And the title of his talk was very similar to mine. I don't remember. It had to do with the origin of life on Earth. He is an expert in the field. And he wrote that book that you see there, The Fifth Miracle, which is a wonderful book about the origin of life. And I recommend it to you. So we were all anxious to see to listen to what Paul Davis had to say about the origin of life. And he stood up in front of us that last day, and he devoted only 1 or 2 minutes to the topic, and then he switched to speak on a bacterium that he was working on that had been isolated from Mono Lake based on its ability to grow in arsenate instead of phosphate. And they published that work later in Science. You may remember this paper because it was very controversial. Okay, but today I will stick to the title of my talk, and I will refer to the origin of life on Earth. Well, this topic is perhaps the biggest pending question in contemporary science and constitutes a challenge to scientists from all fields. In spite of significant progress made in recent decades, mainly in the field of prebiotic chemistry, we are forced to admit that we still do not know much about how matter came to be organized in such a way as to give rise to cellular life. And lack of knowledge, as you all know, gives rise to all sorts of inquiries and controversies. How early did life appear on Earth? Did life emerge in some sort of primordial soup or in hydrothermal vents in the ocean floor? Or was it perhaps in inland hydrothermal fields? Did life instead come from space? Was the first metabolism autotrophic or heterotrophic? Was there an RNA world first, or did metabolism come first? Are we ever going to know the origins of chirality and the origin of the genetic code? But above all, how did inert matter organize to give rise to the first organisms? And at this stage, I would like to say a few words about the transition between inert matter and an identifiable living organism. This is an issue that is very seldom discussed. Indeed, upon reading the literature, one is left with the impression that all that is required to get life going are lipid vesicles and some sort of information polymers that can self-replicate inside them. The first statement I would like to make in this respect is that life entails something more than its material components. It is the organization pattern of the latter, more than the identity, which is critical for life. On the other hand, common sense tells me that entities that are half inert or half alive are difficult to perceive. And accordingly, we must assume that life is something that appears when a certain threshold between inert matter and a living organism is crossed. Now, what do I mean by a living organism? And in this occasion, and at the risk of appearing somewhat chauvinistic, I will make use of the approach offered by the Chilean scientists Humberto Maturana and Francisco Varela, namely autopoiesis. As defined by them, an autopoietic system consists of a network of processes of production in which the function of each component is to participate in the production or transformation of the other component of the network, including the production of an external limit that specifies the domain of the autopoietic organization. I am certainly aware that this definition is incomplete. For example, it does not specify that the system sustains itself autonomously, far from equilibrium. Neither does it mention the property of Darwinian evolution that is so typical of living beings. However, I think it is a good start when attempting to define an organism. And in keeping with this reasoning, I would not consider self-replicating polymers floating in a primordial soup to be alive, not to mention the unlikelihood of this scenario. In contrast, I would definitely consider a self-sustaining single cell that is autonomous in obtaining its energy and nutrients in spite of the changing environment to be a living organism, or more precisely in this case, a microorganism. Having clarified this critical issue, the following step is to try to envision the transition from inert matter to life in the early Earth. Most likely, and this is pure conjecture, primeval life must have been much simpler than extant life. I don't see how inert matter could cross the threshold to life to become at once a microorganism such as Mycoplasma genitalium, the simplest bacterium that can be grown in the lab. This bacterium has about 400 genes. How much simpler can life get? We just don't know, but we can speculate. And in my opinion, nobody has done this exercise better than Shostak, Bartel, and Luisi, whom about 15 years ago proposed the theoretically simplest cell, which is here. According to these authors, this hypothetical minimal cell consists in a vesicle that contains a self-replicating ribozyme that also replicates a second ribozyme that is involved in the formation of the lipid membrane, thus closing the autopoietic circuit. The transition from non-life to a minimal organization of this sort would still be a puzzle. Think, for example, on the provision of activated nucleotides, although perhaps more easy to solve. Since my time this evening is quite short, I have to compress billions of years in 25 minutes that AD allowed me. I will be very specific, and I will refer to a likely scenario for the origin of life. The notion of a primordial soup has been dominant for decades among researchers in the field. This theory was independently proposed by Alexander Oparin in Russia and John Haldane in the UK almost a century ago. This theory states that organic compounds formed on Earth, together with those brought by meteorites, self-organize to give rise to the first heterotrophic microorganisms. I am also including in this slide a photo of the late Stanley Miller, whom with his seminal work in experimental prebiotic chemistry started in 1953, gave support to the theory, of which he was indeed a strong advocate. However, with all due respect to this scientist, I personally don't favor this theory mainly for two reasons. The most important one is that life implies disequilibrium, and the opportunities for disequilibrium offered by the soup are not only limited, but also transient. In addition, most prebiotic chemistry supporting the primordial soup theory has no parsimony with extant life. Indeed, no known forms of life uses compounds such as cyanide, formamide, or cyanamide as substrates for carbon or nitrogen fixation. Nor does any known form of life use UV radiation or lightning as a source of energy. In recent years, on the other hand, the hypothesis of submarine hydrothermal vents has gained considerable attention. Sorry. We must have in mind that there are two types of hydrothermal vents. One of them is represented by the so-called black smokers, discovered by the submersible Alvin in 1977 near the Galapagos. In this case, the seawater percolates through fissures or cracks present in the ocean crust in the vicinity of subduction zones. The water reaches the magma and is heated up to 400 degrees and then reemerges carrying dissolved salts that precipitate when the hot water mixes with the cold ocean water, giving the impression of a chimney. According to Günther Wasterhäuser and others, life started in these black smokers with the formation of organic molecules that thereafter established a surface metabolism with energy deriving from the formation of pyrite, which is a very exergonic reaction. The scheme in the right shows a summary of the reactions that have been reproduced in the laboratory under hydrothermal prebiotic conditions, which include the formation of amino acids, lipids, thioesters, peptide bonds, etc. The main criticism against the black smoker scenario are that prebiotic synthesis of organic compounds relies mainly on carbon monoxide rather than carbon dioxide fixation. The concentration of the former in the Hadean Ocean is presumed to have been negligible. In addition, no known form of life is powered by energy deriving from pyrite formation. And then there are the lost city type hydrothermal vents, discovered more recently in the Atlantic Ocean, although they are present elsewhere. In this case, the water warms up as it percolates the ocean down to the depth of about 5 kilometers without reaching the magma. As the ocean flows underground, it collects the products of a geologic process called serpentinization, which include some hydroxides, hydrogen, methane, and other hydrocarbons. Thereafter, the alkaline fluid, with temperatures in the range of 50 to 90 degrees, mixes with the acidic cold water, leading to the precipitation of carbonates, silicates, clays, and sulfurs of iron and nickel. The chimneys formed are porous and can reach up to 60 meters high, and they are shown at the right in this picture. They make the cover of Nature. You can see the towers of porous mineral there. I mentioned serpentinization. Serpentinization is a geologic process that typically takes place in ultramafic rock. This slide shows representative reactions of the serpentinization process. At high pressure and temperatures of around 150 degrees, olivine reacts with water producing serpentinite, magnesium hydroxide, and hydrogen, among other products. Hydrogen is also produced in other reactions, whereas methane is produced by reduction of CO2, as you can see in the two bottom reactions. Serpentinization is a highly exergonic process. In summary, water oxidizes iron 2 to iron 3. The water is reduced to hydrogen, with its oxygen being retained in the rock as iron oxides. The resulting solution is warm, alkaline, and reducing. And I will repeat that. The resulting solution is warm, alkaline, and reducing. As the alkaline hydrothermal fluid flows through the porous towers, it establishes a natural disequilibrium with the ocean water. As you can see in this cartoon, the resulting natural proton gradient in the left is very similar to the one that is established in the mitochondria or in a bacterial cell in the right, where protons are pumped to the outside. It is this disequilibrium, namely temperature, pH, and redox, which provides the energy that can be harnessed for organic synthesis and for the organization of matter, what makes so attractive the low city type vents as a conceivable scenario for the origin of life. I am now going to refer to the very first step of prebiotic organic synthesis in the lost city type vents, as proposed by Michael Russell and collaborators. This slide depicts the hydrothermal mound in the ocean floor where serpentinization is taking place. So below the ground is the serpentinization process. The overall picture is in very simple terms look like this. There is hydrogen, there is methane being produced by serpentinization, both flowing into the porous rock. There is also CO2 in the ocean water. This CO2 is reduced to carbon monoxide by hydrogen, whereas methane is oxidized to methanol by an oxidant such as nitrate or nitrite, which will thereafter react with carbon monoxide to give acetate. Metals such as nickel and iron that are present in the inorganic walls of the porous mound may play a key role in catalyzing these reactions. The life of methanogens in lost city vents today is basically supported by this type of redox reaction. Let me now tell you more about the transition element sulfides, hydroxides, and oxides that are present in the porous rocks in these vents. With structures, the natural compounds are very similar to prosthetic groups present in extant metalloenzymes. Among them, I would like to highlight, well, you cannot see much from your seats, but you can believe what I say. Among them, I would like to highlight the mineral greigite, it's in B left, whose structure is very similar to the metal prosthetic group of the enzyme acetyl-CoA synthetase, B right, and violarite, C left, similar to the metal prosthetic group of carbon monoxide dehydrogenase, C right. They both contain nickel in addition to iron and sulfur. As I just said, these and other minerals present in the rocks are proposed to have played a critical role as early catalysts in prebiotic reactions. This is not for you to read, but it shows a long list of representative reactions conducted under prebiotic conditions at alkaline pH that might be relevant to early metabolism in the hydrothermal mound. The list includes the formation of sugars, fixation of CO2 and nitrogen, oligomerization of amino acids and nucleotides, vesicle formation, etc. Most of these reactions are catalyzed by metal centers based on reaction of this, such as this. Russell, Martin, Lane, Nietzsche, and collaborators have proposed that the first organisms were autotrophs and used the acetyl-CoA or Wood-Lundahl pathway to fix CO2. This ancient pathway has a strong resemblance with the hypothetical prebiotic chemistry in the hydrothermal mound and fits well with several reactions listed in this slide. This is a very simplified scheme of the methanogenic left and acetogenic right Wood-Lundahl pathways. Steps restricted to bacteria are marked in bright blue color, as you can see in the right, whereas those found only in archaea are shown in violet. Dark blue stands for reactions and enzymes observed in both prokaryotic domains, which suggests that this common pathway is even more primitive. CODH stands for carbon dioxide dehydrogenase, which catalyzes the reduction of CO2 to CO, whereas ACS stands for acetyl-CoA synthetase, which catalyzes the formation of CC bond and thioester bonds. Metal cofactors in CODH are molybdenum, copper, and iron, whereas acetyl-CoA synthetase has a nickel-iron cluster. Both pathways contain a proton motive force generating step. This is something very unique. This CO2 fixation pathway generates energy through proton motive force. And the two steps are boxed in red in either pathway. Let me now zoom. No, I will skip. I will just give you the main reasons for Russell, Martin, and collaborators to give support to the Wood-Lundahl pathway in the early autotrophic metabolism, and these are the following. It is the only CO2 fixation pathway that is present in both archaea, methanogens, and bacteria, acetogens. It is the only exergonic CO2 fixation pathway. Again, this is something very unique, an exergonic CO2 fixation pathway. In both domains, the energy is used to generate a chemiosmotic gradient. Proteins in the pathways in both have numerous prosthetic groups containing iron, nickel, and sulfur. It has parsimony with hydrothermal vents prebiotic chemistry. And finally, microbial methanogenesis was already functioning 3.5 billion years ago. Interestingly, Bill Martin recently published a paper in which he analyzed 6.1 million protein-coding genes from sequenced prokaryotic genomes in order to reconstruct the microbial ecology of LUCA, the last universal common ancestor. According to Martin and collaborators, LUCA was anaerobic, thermophilic, fixed CO2 using the Wood-Lundahl pathway, and was also able to fix nitrogen. In addition, according to this study, LUCA's biochemistry was replete with iron-sulfur clusters, transition metals, and radical reaction mechanisms. Bill Martin was here in this room a couple of years ago, and he gave a talk about his work on a meeting that we held on big data. Another quite attractive feature of this theory is that the porous rocks provide a proper scaffold for the self-organization of the first organic molecules. Again, you will not see much here, but you can imagine what I want to say. In the left, you can see all the products from serpentinization flowing through this porous rock, and you have the microcompartments of the porous rock that are receiving these products, namely methane, hydrogen, and so on. Thus, the sustained flow of serpentinization fluid across the mineral compartments establishes a permanent redox pH and temperature gradient with a persistent source of electrons for a copious prebiotic chemistry in catalytic surfaces of iron sulfide containing other catalytic metals. Thus, an initial step of prebiotic chemistry could have been followed by a subsequent step of an RNA world, perhaps, then a DNA world with the establishment of LUCA, which is still not free living. So in this scheme, LUCA is still attached to the porous rock. The following step might have been the development of a cell membrane biochemistry for archaea and bacteria, which is different, to end up with cells that are able to detach from the mound and become free-living microorganisms. This is, of course, just a model, a proposition. There are innumerable questions still pending, but still, it is perhaps the best hypothesis we have today for the origin of organismic life. I still have some time. Well, Nick Lane, whom I have mentioned several times, and one of the most enthusiastic supporters of this theory, recently attempted to respond a response to the old question regarding whether the emergence of life is inevitable or a fluke. And please allow me to quote this short text published in New Scientist because it is highly suggestive. And I can read it for you. He says, I am the first to admit there are many gaps to fill in, many steps between an electrochemical reactor that produces organic molecules and a living, breathing cell. But consider the bigger picture for a moment. The origin of life needs a very short shopping list: rock, water, and CO2. Water and olivine are among the most abundant substances in the universe. Many planetary atmospheres in the solar system are rich in CO2, suggesting that it is common too. Serpentinization is a spontaneous reaction and should happen on a large scale on any wet, rocky planet. From this perspective, the universe should be teeming with simple cells. Life may indeed be inevitable whenever the conditions are right. It's hardly surprising that life on Earth seems to have begun almost as soon as it could. And precisely, and this is my last slide, regarding the possibility that serpentinization may be a common process in the universe, let me bring to your attention a recent finding in the Saturn moon Enceladus. As you may know, Enceladus has a rocky core completely covered by liquid water, which is frozen on the surface because of the temperature. The Cassini spacecraft discovered large plumes of water vapor erupting from the icy crust and has recently detected hydrogen gas in these plumes, which, according to this report published in Science, most likely this hydrogen arises from hydrothermal reactions in the ocean floor, and there must be some hydrothermal activity there because the water is liquid. The presence of liquid water in this moon constitutes an additional evidence for the hydrothermal activity. This is indeed a very exciting finding, and we wouldn't be surprised if in the near future evidence for microbial life is found in this particular moon and elsewhere in the universe. Thank you very much for your attention. I am really glad that I had enough time.